Good day, this is the Professor, drwillphd, PhD. Today's Monday, August 15, 2022. It is 10:02 and 7 seconds, a.m. Eastern Standard Time. And today's research, mapping tumors. So let's hop to it here. Genetic mapping of tumors reveals how cancers grow. Date August 10, 2022. Source, University of Oxford. The researchers have found that individual prostate tumors contain previously unknown range of genetic variation. Understanding which cells give rise to which areas of cancer can improve our understanding of how a tumor has grown and developed, including how it has changed genetically over time. And this has been made possible using a new technique called spatial transcriptomics, which allows scientists to see what genetic changes take place without breaking up the tissue they are looking at. And this adds a new dimension, which researchers have now used to reveal which cells have mutated and where within the ecosystem of an organ. The current techniques for studying the genetics of cells within tumors involve taking a sample from the cancerous area and analyzing the DNA of those cells. The problem is that many cancers, such as prostate cancer, are three-dimensional, which means that any one sample would only give a small snapshot of the tumor. The researchers used spatial transcriptomics to create a cross-sectional map of a whole prostate, including areas of healthy and cancerous cells. By grouping cells accordingly to similar genetic identity, they were surprised to see areas of supposedly healthy tissue and already had many of the genetic characteristics of cancer. And this finding was surprising because of both the genetic variability within the tissue as well as a large number of cells that would be considered healthy but which contain mutations usually identified with cancerous cells. The prostate tissue is three-dimensional, and like most organs that can develop cancer, we still have much to learn about what cellular changes cause cancer and where it starts. One thing we are fairly confident of is that it starts with genetic mutations. Researchers have never had this level of resolution available before, and this new approach reveals some surprising results. For example, researchers have found that many of the copy number events that previously thought to be linked specifically to cancer are actually already present in benign tissue. And this has big implications for diagnosis and also potentially for deciding which bits of a cancer need treating. Mapping thousands of tissue regions in a single experiment is an unprecedented approach to deconvolute the heterogeneity of tumors and their microenvironment. And this high-resolution view impacts our way of addressing complex ecosystems, such as cancer. The possibility to identify early events is particularly exciting going forward. Furthermore, the researchers analyzed more than 150,000 regions and 3 prostates, 2 breast cancers, some skin, a lymph node, and some brain tissue, and developed an algorithm to track groups of cells with similar genetic changes, clones, and their precise location. And this approach enabled them to zoom right in from visible tissue through microscopic, multicellular structures and right into the genes themselves, while keeping hold of the overall landscape of tissue. Good day, this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.